Hi everyone, welcome to the classes of 𝗏𝗏 where we deal with the microbiology. In microbiology, our sequence of teaching will be, first of all, we will discuss general microbiology, then we'll come on to bacteriology, then mycology, that is the study of fungus, then virology, the study of viruses, parasitology, and in the end we'll deal with some miscellaneous topics. There will be some surplus videos added, the link of which will be provided in the description along with each video so that you can use it in order to enhance your knowledge. Let's quickly start with the general microbiology. In general microbiology, we'll try to sum up in 11 classes. This is class 1 and we are going to discuss about the cell wall. Mind you, bacteria is having a beautiful cell wall, and in order to study cell wall, we will use these headings. First of all, we'll do the introduction. Next is the function of the cell wall, then the structure of the cell wall, then the cell membrane. We'll talk a little about the cell membrane also. Then we'll discuss some associated parts, and in the end, I'll teach you about the applied microbiological aspects for the cell wall. First of all, introduction. Please understand that bacteria has different shapes. Some are spherical, then those spherical shaped bacteria, we call them as cocci. In cocci, you have diplococci, where the cells are joined with each other. So, two joined cells are called as diplococci. Next comes cocobacillus. Now, what is the meaning of word bacillus? Bacillus means it is rod shaped. Bacillus means it is, okay, I'll write it here. Bacilli means it is rod shaped. Okay, so what will be the meaning of word cocobacillus? That means it is spherical as well as it is cylindrical. It is spherical as well as it is rod shaped. So cocobacilli, right? So please understand what are the examples of diplococcus. Diplococcus means two circular ones. Diplococcus means two spherical ones joined together. So the example is going to be coccus, Streptococcus pneumoniae, meningococcus. Streptococcus, meningococcus. Coming on to the next part, that is cocobacilli. So, the mnemonic to remember cocobacilli is high RCB. So, now this is the time for RCB. Just like after putting so much efforts, just like after facing so many defeats, RCB is the winner finally. This is going to be your story, my dear friends. Remember Thomas Alva Edison, he tried so many times and it was his 101st time when he was able to complete his discovery. So similarly, RCB rocked and it's the time for you to rock with us in the 𝗏𝗏. Coming back to the topic, the mnemonic to remember high RCB. Yeah, so mnemonic to remember cocobacilli is high RCB. What does that mean? High means Haemophilus influenza. High means Haemophilus influenza. What is the meaning of RCB then? C for coco, B for bacillus, and the example from C is chlamydia. So cocobacilli has got two examples, that is Haemophilus influenza and chlamydia. So I've written Haemophilus influenza and chlamydia. So cocobacilli can be described as intermediate shape between the coccus and the bacillus. Coming on to the two very important questions which they are fond of asking, be it any examination of viva, they are fond of asking. Streptococci and Staphylococcus. How I remember is, okay, first of all let me tell you, Streptococcus is something that is there in chains. Streptococci prefer to be in chains, and Staphylococcus is something that prefer to be in cluster just like grapes. Okay, so how to remember? Phyle, the word phyle is coming in Staphylococcus. Phyle in literal sense means love, and everybody has got a beautiful love for grapes. So Staphylococcus loves to be in cluster just like grapes bunch, the bunch of grapes, and Streptococcus loves chain. So Streptococcus is in chain. Okay. Also there are some other shapes just like rods. I have told you rods. Now rod shaped bacteria, they are called as bacillus, and Bacillus anthracis is one of the example. I'll correct the spelling. Bacillus anthracis is the example for rots. Then you have got spiral shaped, this diagram. Then you have got spiral shaped bacteria, and the example is going to be spirulina. Coming on to the next important part, what is bag? So B and A, can you see here? I've just written one G. So Bacillus anthracis has got glutamic acid. Bacillus anthracis has got a lot of glutamic acid, and because Bacillus anthracis has got a lot of glutamic acid, the G is resembling with C. So there is a structure with Bacillus anthracis that is called as capsule, and the capsule of Bacillus anthracis is made up of glutamic acid. Now, why did I mention it here? Because when I will teach you about the capsule, I am definitely going to ask you that the Bacillus anthracis capsule is made up of what? Please remember the mnemonic bag, and then you will tell me that it is made up of glutamic acid. It is made up of glutamic acid. Rest all have a polysaccharide made up of, rest all have got a capsule made up of polysaccharide. So, intentionally I have brought this topic over here in order to make the layers clear. First of all, we have got cell membrane. After cell membrane, we have got cell wall. And finally, we have got the third layer, that is capsule. Okay. So, I think the confusion of the layers is clear, and a beautiful example of Bacillus anthracis known by mnemonic bag is G, that is made up of glutamic acid. Okay. Coming on to the next heading, that is what is the function and composition. So, we are done with the introductory part. Now, we will discuss about the function and the composition. So, please understand about the function and composition. You have a mnemonic that is vocal. For example, you can remember my own function. What is my own function? Sitting in the chair and talking and talking and talking. So, function, vocal, vocal, function. Mnemonic to remember the functions of cell wall is vocal. V for virulence, V for virulence, O for osmotic stability. Cell wall is known to provide virulence. Cell wall is known to provide osmotic stability. Cell wall has got peptidoglycan. So, V for virulence, O for osmotic stability, C for can, peptidoglycan, that is responsible for providing rigidity. That is responsible for providing rigidity. A for antigenic. A for antigenic. Sir, earlier you told us V for vocal. Now A for antigenic. Aren't both these same? No. Let's discuss what is the difference between virulence and what do you mean by antigenicity. See here, virulence is because of endotoxins being produced. Virulence is because of endotoxins being produced, and these are produced because of the cell wall only. Let me discuss it here. So, gram-negative bacteria has got lipid in their lipid A in their LPS layer of cell wall. Just learn this, don't be panicked, don't feel that you are not aware of whatever I am teaching. Hold on. So, you know that in microbiology, the very important thing is gram staining. So, there are some bacteria which take up the stain and become gram positive. There are some bacteria which don't take up the stain and become gram negative. So, gram negative bacteria, they have got lipid A in the LPS layer of the cell wall. So, when we will discuss the layers of the cell wall, I'll teach you what is LPS layer. So, in the LPS layer of the cell wall, you have got a lipid A, and that is responsible for formation of endotoxin, right? That is responsible for formation of exotoxins or endotoxins. For that matter, you can remember toxins, okay? So, that is responsible for making the toxins, and this is responsible for the virulence. O for osmotic stability. C for can, that is peptidoglycan, responsible for giving the rigidity, and A is antigenicity. Antigenesis means antibody generation. So, the cell wall is responsible for antibody generation. The cell wall is responsible for generation of toxins, that is responsible for giving the virulence to the bacteria. And A and L I have intentionally kept side by side because the antigenic property is coming because of lipoteichoic acid. Lipoteichoic acid. And teichoic, can you see this tick? And if you are marking your checklist, done, that gives you positivity. That means this lipoteichoic acid is giving you positivity. What does it signify, or why I am telling you like this? Because lipoteichoic acid is present in the gram-positive bacteria. It is present where? In the gram-positive bacteria. Okay. So today, remember, you have to stand on the wall and speak in order to be functional. What does that mean? Stand on the wall, that means I am talking of the cell wall, and speak, that means become vocal. So VOCAL, in order to function, that means it is the mnemonic for functions of, yes, functions of what? Functions of cell wall, right? Now coming on to an important part, peptidoglycan. Remember, peptido and glycan. Peptido means peptide, that means protein, and glycan. So both of them, peptido and glycan, they have got linked by the enzyme which is called as transpeptidase enzyme, which is called as what? Transpeptidase enzyme. Transpeptidase enzyme is something that is inhibited by beta-lactam group of antibiotics. Transpeptidase is something that is inhibited by beta-lactam group of antibiotics, and once this beta-lactam group of antibiotics act on the bacteria, the peptidoglycan is going to get vanished. The bacteria will no more be rigid and the survival will not be there. Bacteria is going to die. That is why beta-lactams are used as antibacterials. Now your task is to comment in the section below that what are the examples of beta-lactams, because if I'll tell you, the pharmacology faculty will come and beat me like anything. Sir, why are you snatching my work? Huh? So just let me know in the end, in the comment section, what are the examples of beta-lactams. Mnemonic I'll give you, BBC news channel, very famous. Okay, so BBC, very famous. So you will comment down and tell me. Okay, and in order to remember that beta-lactams act on the transpeptidase, I have got a mnemonic. Can you see, this is P, and if you add one more circle, semicircle, that becomes B. So beta-lactams are going to inhibit the peptidoglycan by inhibiting the transpeptidase enzyme. Coming on to the structure. So see here, if you carefully look at the peptidoglycan, you have got a molecule called as NAG, you have got another molecule called as NAM. So NAG and NAM, both of them are there, and the NAM, okay, full forms are not necessary at UG level, so I will not teach it. See it, NAG and NAM. So NAM has got 4 amino acids, L-alanine, D-glutamine, L-lysine, D-alanine. So LDLD, LDLD. Remember, this is not DL, that's not Delhi, this is LDLD. Okay, so LDLD. So L for L-alanine, D last alanine. So first and last are alanine. Remaining ones are 1D and L. So L is there, so you can remember another L, that is lysine. The remaining one you have to remember, that is glutamine. Remaining one you have to glue into your brain, that it is glutamine. Okay. Coming on to the next part, what is the difference between the gram-positive and gram-negative bacteria? Remember, gram-positive has a lot of P's. So gram-positive has got a lot of P's. So what are the P's? P for peptidoglycan, that is thick in case of gram-positive. Next P is periplasmic space, that is also having the P in it. Third P is porins. Third P is porins. But if I say that porins are present in gram-positive, that's going to be a wrong state. Okay. So see, I told you 3 P's. One P is peptidoglycan, other P is periplasmic space, and third P is porins. So peptidoglycan is there and it is very thick in case of gram-positive bacteria. If I talk about the periplasmic space, it is also very thick in gram-positive bacteria, whereas it is thin in case of gram-negative ones. But remember, porins. Pore is just like a gap, and gap creates negativity. my and my romantic partner, if we are living at gap, it is going to create a negative impact. That's why porins are there in gram-negative bacteria. Okay, they are present in gram-negative, exclusively present in gram-negative. So see here, void has negativity. Okay, so remember, porins are present on the outer membrane. Okay, porins are present on the outer membrane. So outer membrane, porins, both of them are the feature of gram-negative bacteria. Okay, so my name you will remember, right? P. So P for positive, P for PPP, but not the last P. And I love to eat oats. So whenever you are welcoming me to your home, please prepare oats. I love oats. Okay. So OATS, oats is the mnemonic. Next mnemonic. O means outer membrane, has got porins in the gram-negative ones. Coming on to A and T. So A for aromatic amino acid and T for teichoic acid. So I've already told you teichoic means tick, and if there are a lot of ticks, tick, then it is going to give you positivity. So it is present in the gram-positive bacteria, whereas the remaining one, A, aromatic, is present in the gram-negative bacteria. Okay, gram-negative bacteria. And if you see, negative is having A in it, whereas positive is not going to have A in it. So what this A signifies? A for aromatic amino acids. So this is all about this beautiful table. Okay. Now let's quickly understand very important thing, that LPS, lipopolysaccharide. What does it do? Lipopolysaccharide has got a lot of functions we'll discuss, but the very important function is that it induces TNF-alpha. It releases interleukin 1. Now, what are these molecules and what is the significance? Pathology ma'am will tell you. If I will tell you, pathology ma'am will come. She will take me and hit me like anything. So, LPS is responsible for inducing TNF-alpha, interleukin 1. And both of them are responsible for causing fever. Responsible for causing what? Fever. And in the cell wall, there is a polysaccharide called as 𝗲𝗵 that is antigen. Okay, you got it? Great. So, just for your revision, the outer cell membrane has got porins, right, in the gram-negative bacteria. Now, what is the function of that porin? So, supposedly this is a pore. What is the function of that pore? It allows the transmission of hydrophilic molecule. So, my dear friends, what happens is, see, the cell wall is hydrophobic. Cell wall is hydrophobic. So, it can allow the incoming of hydrophobic molecules. That's okay. But porins allow the incoming of hydrophilic molecules such as penicillins. Okay, such as penicillins, which are ultimately going to kill the bacteria. But then penicillins are going to take the entry with the help of porins only. See here, a very important previous year question. The structure that makes the bacteria drug resistant is drug resistant. What does that mean? That you are giving the drug, but the drug is not active. So, the structures that make the bacteria drug resistant is called as periplasmic space. Why? Because it has got some enzymes that are going to make the bacteria drug resistant. For example, I told you a drug which is called as beta-lactam, and the beta-lactams are known to kill the cell wall. But bacteria are smart and intelligent. What they will do? In the cell wall, they will have a periplasmic space. I have told you. In the periplasmic space, they will put beta-lactamase. That is something that is going to kill the beta-lactam. And once the beta-lactam is killed, obviously, the bacteria is going to be resistant. Bacteria will not be killed by beta-lactam. I'll summarize it for you. Beta-lactams normally kill the cell wall, but the bacteria in their cell wall has now started keeping periplasmic space, and in this periplasmic space, the bacteria has beta-lactamase. It is going to kill the beta-lactam. Once the beta-lactam is killed, obviously, the bacteria will be labeled as resistant to beta-lactams. Okay, right? And the next question is L forms. So, for example, I'll give you an analogy. For example, I'm wearing this black cloth, and if I wear this black cloth and go to attend a funeral, people will say, "Oh, please go away," or somebody some people might gather and beat me, hit me also. So what? I will remove this black cloth in order to get saved from the mob attacking me, right? So similarly, in order to get saved, sometimes bacteria sheds off the clothes, bacteria sheds off the cell wall, and the bacteria without a cell wall then will be called as L form. Which form? L form. Okay, it will be called as L form. Okay, so this was a question which was asked. Now, teaching you in detail what is the LPS layer of gram-negative bacteria. So, can you see here? This is the LPS layer which has got three parts, or I should say two parts. One is the outer core, other is the inner core, and on the outer core has the O antigen. I've already told you that O antigen is responsible for the antigenicity in the cell wall. And if you remember the mnemonic local, A for antigenicity, right? Because of lipopolysaccharide acid also, and because of O antigen as well. So outer core, inner core, right? Whereas in flagella is also responsible for antigenicity, but flagellar antigenicity is because of H antigen, and the mnemonic is 𝗵𝗵𝗵𝗺. So can you see here, my dear friends, F and H coming together? So flagella having H antigen. So O antigen we have talked about, present in the outer core, and then you have got an inner core where you have got a lipid A, and then lipid A is responsible for what? We have already discussed, for the production of toxin. Okay, so quickly summarizing, O antigen in the outer core for the antigenicity of the cell wall, H antigen, flagella, 𝗵𝗵𝗵𝗺, whereas inner core is having lipid A. Lipid A is for toxin production. Coming on to a small little discussion about the cytoplasmic membrane. So remember, mem is just like mum, just like your mum. Cell membrane, cytoplasmic membrane is also mum. So mum does what? Mum is the precursor. Okay, so she is a precursor for the cell wall synthesis. She is the most energetic member of your family. So cell membrane does the energy generation by oxidative phosphorylation, and it is responsible for active transport of molecules into the cell. Because she is active, so what she'll do? She'll transport the molecules actively. Now, coming on to the very interesting story of Joker. Who Joker? For a mama, every child is a Joker. But for a plant kingdom, the Joker is not child. The Joker is Mycoplasma. Okay? The Mycoplasma is called as Joker of the plant kingdom. Why? Because it does not have cell wall. It does not have cell wall. So if you will not wear clothes properly and go outside, people will say, oh, see the Joker. So if Mycoplasma is not wearing the cell wall properly, everybody is saying, Mycoplasma, you are a Joker. Okay? So you can comment down in the comment section and tell me who is the Joker of your friend group. Huh? So let's do a quick revision, that the periplasm is the space between the inner and outer membrane in the gram-negative bacteria. Okay, and peptidoglycan is located within the periplasmic space in the gram-negative bacteria, and beta-lactamase are also located in the periplasmic space in the gram-negative bacteria we have discussed. Coming on to the cytoplasm and the ribosome. Okay, so cytoplasm. So see here, bacterial cytoplasm has two distinct regions on the microscopy. Can you see here? Yeah, see here. So on the microscopy, they have got two distinct regions. Can you see this one? This is the nucleoid region where we have got the circular DNA. So this is the circular DNA. So this is the nucleoid region responsible for the genetic. Whereas the other part of cytoplasm is this blue part, and this blue part is the amorphous part of the cytoplasm. Okay. So I've told you that it has got two parts, amorphous matrix and the nucleoid region. Nucleoid region has the DNA, whereas amorphous matrix is present in the cytoplasm. So bacterial cytoplasm has two distinct regions, amorphous matrix and the nucleoid region which contains DNA. Got it? Coming on to the next part, ribosomes. So remember, ribosomes has got two subunits, that is 30S and 50S. Now as all the biologists all the medicine people, all of us doctors are very famous for a beautiful maths. Similarly, you are not supposed to apply your maths here. 50 plus 30 is not 80, it is 70. It is not 80, it is 70. So, in total, bacteria has got 70s ribosomes with 2 subunits, 30s and 50s, okay, which are responsible, just like any other ribosome, it is responsible for the protein synthesis. But what is important? Important is that you have got 2 group of antibiotics and 2 more group of antibiotics. So basically 4 group of antibiotics that are going to hit and kill the ribosome. So ribosome was doing what? Ribosome was doing the protein synthesis, and proteins were essential for the survival of the bacteria. But what happened is somebody came, killed the ribosomes. Oh my God, what to do? Then came microbiologists, the Bahubalis, and identified that, okay, which ribosomes are you killing? Which ribosomes are you killing? So, coming in the class from the role of Bahubali, and let's try to understand that where these antibodies, the antibiotics are killing the ribosomes. So, you have got 𝗲𝗏, 𝗲𝗏𝗏𝗏, T for T, that is tetracycline, A for AMG, that is aminoglycosides. They are going to act on the 30S ribosomes, whereas 50 is a big number, macro. So, macrolide group of antibiotics are going to come and kill the 50S ribosomes, whereas Ram, Lord Ram is also a big man. So, bigger unit, 50S, is being killed by Ramji himself, 𝗲𝗵𝗿𝗺. Okay. So, ribosomes have got 2 units, 30S, 50S. 30S will be killed by T and A, tetracycline, aminoglycoside. 50S will be killed by big people, macrolide, Ramji, Lord Ramji, 𝗲𝗵𝗿𝗺. You got it? Got it? Good. And remember, the bacterial DNA, which I have already shown you, we will discuss it in the subsequent sessions. So, can you see here, this is the nucleoid, the circular DNA, these are the ribosomes, this is the cytoplasm. Here if you see, this is the plasma membrane, then the cell wall and the capsule, which we have already discussed. Then coming on to the glycocalyx. What is glycocalyx? Glycocalyx is a loose network of polysaccharide. So, it's a network of polysaccharide material that mediates the adherence to the surface. So, glycocalyx leads to adherence to the surface. Okay, and I always say G and C look similar. So, glycocalyx is the material which is responsible for causing the catheter associated UTI. What does that mean? Okay, let me take you to my internship days. Once, this was the very first time when I started doing the catheterization to patients, and one day my ma'am said, "Oh, Parv," yes ma'am, "please clean your hands properly." I said, "What happened, ma'am?" She said, "Do you know, if you are not keeping hygiene, it will lead to 𝗲𝗏𝗏𝗏." I was not aware what is 𝗲𝗏𝗏𝗏. That is catheter associated UTI. I thought, ma'am, how the urine is going to come? It is going to wash away all the bacteria, even if there will be bacteria inside the catheter. How can bacteria stick to the wall? Then madam said, it has got a glycocalyx. The glycocalyx will allow the bacteria to adhere to the surface. So, glycocalyx mediates the adherence to the surface, and that is why it is responsible for causing the 𝗲𝗏𝗏𝗏, that is catheter associated UTI, that is urinary tract infection. I think I have written it below. Yeah, C I have written. C and G you can remember. Coming on to the capsule. So, I have told you capsule, bag is the mnemonic, Bacillus anthracis made up of glutamic acid, whereas all others are made up of polysaccharides, which I have written over here. And remember, whenever the word capsule comes, you have to recite a poem. AVSM has capsule. I am a little bit good at singing skills. Someday I'll sing for you also. But then AVSM have capsule. What is AVSM? A for antiphagocytic, V for virulence, S for serotype, and M I leave for you. You will tell me in the comment what is M, or otherwise we will discuss in the class of capsule. But 𝗵𝗵, yes, 𝗵SM has capsule. Okay, you have to remember this. So A for antiphagocytic, V for virulence, S for serotype. Okay, so capsule is antiphagocytic. Capsule does not allow the phagocytosis of bacteria. V for virulence, it is responsible for the virulence of the bacteria. S for serotype. So bacteria has got different serotypes, okay, because of the presence of capsule only. Okay, so this is all about today's class. Thank you so much. We'll see you in the next class, and the comment section is always open for your blessings, for your love, support, and definitely the feedbacks. Thank you so much.